Now let's take a look at how we actually conduct variant calling on some data. So first we will go to the T-bioinfo platform where we will find an areas of analysis section. So in this areas of analysis, you can see there are many different sections related to transcriptomic, genomic, metagenomic, and other kinds of data. Here we will look for mutation variant section. If you hover over the eye icon, you will get more information. You can see that this one is going to be appropriate for exome sequencing data. If we go within this section and just click on the title, we will be prompted to upload data. This data can come from NCBI, from our computer, or from the FTP account if you have a research account with us. But for now, we will use the demo option. So we will click on this drop down, and here let's select this last example for breast cancer mutation. So let's click on this link, and what you should see is an interface for the pipeline builder. You will only get an option to select something that is highlighted, and you can see that the first thing that is highlighted is the start button. Here you will have a little bit of information about what this pipeline is all about, and it's going to be about finding variants in this data. Now, once we've started, we have our next option to select, and that is Bowtie 2. In each one of these steps, you will get information about the algorithm, and briefly what Bowtie 2 does is it hashes all of the sequences of the reads that we have in our FASTQ file, arranges them so that it is quick to find the most appropriate position on the reference genome based on the matching sequence. Now you can also click on input files and you can see what we have to upload to be able to use this section. And this includes a FASTQ file for each one of the samples that we have, and we have several samples here. It will also have a link to the reference genome. This is important because you can see that each one of the files that we originally uploaded is in SRA format. SRA is a format to compress raw FASTQ files because they are so large. After we've mapped our reads onto the reference genome, what we get is a BAM file or a SAM file that contain the information about each read and the position on the genome where this read aligns to. We can visualize this data. So let's click on visualization. And here you can look at input files. And you can see that we have the FASTQ files, but also these SAM files. And like I said, FASTQ files are reads, just their sequences in random order. And the SAM file is when they are organized also by position on the reference genome. So after we have visualized the data, we also want to determine whether any variants can be called on the collection of reads that fall in a specific region. And for that, we will use FreeBayes. FreeBayes is a haplotype-based variant detector, and it's a tool that works really fast. It takes in the BAM files, which is already the reads that have been aligned to the reference genome, and looks at how many reads contain a specific change from the reference to be able to call that a variant. Now, the output file, if you click here, is going to be a VCF. And that is the VCF that we will later work with to determine whether any of the variants are actually clinically relevant. Now let's click on End. Okay. After your page reloads, you will click here on the visualization button. The visualization opens up in JBrowse. And JBrowse is a software that allows you to visualize the SAM file and the index genome and actually look through the different output files that we have in the pipeline. So here down below, you have what's called mapping statistics. It tells you how many of the reads were paired, how many aligned correctly, and how many aligned incorrectly. And also gives you an overall alignment rate. So you can see that for each sample, we have an alignment rate, and it's very good. It's over 97%. So that means that the alignment worked really well. One of the samples is a little bit below that, 96.76, but that is still pretty good. Now here on the side, you will have multiple tracks. You can open these tracks. Let's just take one sample. And then you can also look at the VCF file for variant calling, which means that it will only show us whether a certain position is a variant. As you scroll through, you will find that some segments have no reads at all, and others will have some reads. So here, each bar like this represents a read. So let's try searching for a gene. One important gene in cancer is P53. P53 is the name of the protein, and the name of the gene is TP53. As you type it in here, you can then select the gene. And now you will see that this particular gene has a lot of reads aligned to the reference. So hovering over each position, you will have the count of total reads that are actually mapped to this region. Down here, you will have your gene in the GTF file. So we'll display all of the transcripts that are available for this gene. The thick bars are exons, and the lines are introns, or non-coding regions. You can see that approximately the accumulation of reads corresponds to the exons that we have, even though it is not precise. In the middle, you can see the information about each one of the variants, and you can see that there are different kinds. Single nucleotide variant, substitution, insertion. Here you will have an information about this specific variant that is identified. For example, for this insertion, you can see that TACAA turned into TACAA-CAA. So we have an additional 3 nucleotides that were inserted in this position.